Speaking of happy to be here, I'm ready to bring out our first guest of the night. She is a neurogenomist working at the University of Helsinki in the Department of Biosciences, and she's going to talk about anxiety disorders. And so if you have an anxiety disorder, you are welcome here. So don't panic! Please welcome to the stage, Iiris Hovatta.
So good evening, everybody. It's so great to see so many of you here today. So I'm here to tell you today how we can use genetics to understand the neurobiological basis of behavior. So my lab at the University of Helsinki is studying the genetic and neurobiological basis of anxiety disorders. First, I want you to imagine that it's a hot, sunny day. You're spending time with your friends at the beach. And imagine the bodily sensations that you have at this moment. The cool water feels very relaxing on your skin, right? But then, how do your bodily sensations change if you think that there's a white shark beside you? So what kind of bodily sensations do you have in this kind of situation? Racing heart, sweaty palms, dry mouth, right? So we all know what anxiety is because it's a normal response to a threatening situations. But in some individuals, these responses are prolonged, exaggerated, and they disturb daily life. And we can talk about anxiety disorders. So these are a group of diseases, and the common feature is heightened anxiety levels. And then they can be divided into different subcategories based on specific features. For example, in panic disorder, the anxiety comes in attacks that are very intense and last between 2 and 30 minutes. People with social phobia would be terrified if they had to switch places with me, or if they have to go into a social situation with unfamiliar people. Most common specific phobias are fear of heights, spiders, snakes, blood, flying, and so forth. Most of the people can cope very well with their disease, but it can also be very debilitating. There are individuals who can't leave their homes in the fear of getting a panic attack, for example. So anxiety disorders are the second most common brain diseases after migraine, and about 5% of the people in Finland suffer from them every year. So what happens in the brain when we see a shark or encounter another anxious stimulus? So the sensory information, the sight, smell, or sound, are received in the brain region called the thalamus. And the thalamus sends signals to another brain region called the amygdala. These are small brain regions on both sides of our brain, and they are the centers of the processing of emotional regulation and emotional information in the brain. And amygdala is highly activated when we are in an anxious situation. And the amygdala talks to many other brain regions. It talks with hypothalamus, that's responsible for the physiological manifestations of anxiety, the racing heart, sweaty palms, and dry mouth, that are all due to the activation of the autonomous nervous system. Amygdala also sends signals to the motor areas of the brain that prepare us to escape if needed. It also talks with the frontal cortex, which is responsible for the cognitive processing of the threat. Is this a real threat? How should I respond? How did I respond in a similar situation previously, and how did that go? Now, it's very important to have the right level of alertness in our brain so that we can respond quickly if there is a threat, and respond with an action that's in correct magnitude, taking into account the threat. But then again, it's important to go back into the normal level of alertness, because if we have very heightened level of alertness all the time, it takes a lot of our resources and energy. And in individuals with anxiety disorders, this balance is disturbed. So about half of the anxiety disorder patients benefit from current medications or cognitive behavioral therapy. So there's a great need for better medications or better treatment practices and more personalized forms of treatment that take into account the personal qualities of each patient. And this is where genetics comes into picture. So anxiety disorders are partly genetic. They run in families, just like many other common diseases like cardiovascular diseases or diabetes. And if we can identify a gene that predisposes to anxiety disorders, we immediately know which protein this gene codes for. And then we can study the function of that protein in the cell, in the brain, and understand what happens on the molecular level in the brain when we get anxious. And this allows us to design better drugs that target the specific molecular problem in the brain. So how we do this in practice is that we collect DNA samples from thousands of individuals who suffer from anxiety and matched healthy control individuals. And we compare their genomes, the DNA molecule that the genes are made of. And we look for genes or gene forms that are more common in anxiety disorder patients than in controls. Now, it's important to understand that there's no one anxiety gene that makes you have an anxiety disorder, but it's a combination of tens or hundreds of genes that each have a small effect and in combination affect our susceptibility to get an anxiety disorder. So there are a lot of anxiety disorder patients who do not have one particular susceptibility gene, and there are also some healthy individuals who will carry the anxiety susceptibility gene, but they are not affected because they don't have these other susceptibility genes. So my lab and other groups, we have identified one such gene that predisposes to panic disorder, and it's called the neuropeptide S receptor. And so there are different forms of this receptor depending on which kind of gene we have inherited from our parents. And some individuals have a receptor that is activated more strongly in response to seeing a white shark or another anxious stimulus. And other people have a form of this receptor that does not activate very strongly in response to anxious stimuli. And this activation of amygdala is directly proportional to how much or how threatening we perceive this threat. And in general, patients with anxiety disorders have much stronger activation of amygdala in response to threatening situations. So in this way, our genes can affect how we behave. And this is just one example how genetic research has actually increased our understanding of neurobiology of panic disorder. And now there's a lot of research going on around this neuropeptide S system to see whether it can be used as a drug target for anxiety disorders. And we, of course, first have to understand completely what this system does in the brain before any drugs can be developed. So this is a great time to be a geneticist. We now have tools and methods to study the whole human genome. And we are beginning to understand how variation in different genes and in our genome affect susceptibilities to diseases and our phenotypes. So the genome gives us a handbook of the foundation of of our characteristics. And then environmental factors shape us into who we are and what kind of diseases we will get. And by studying the interaction of genes and environmental factors, we hope to better understand how diseases develop and thereby develop better treatments for various different diseases, such as anxiety disorders. Thank you.